Hello everyone, I am Sadhna More from Department of Microbiology KSKW College Sidco. Today I am going to present the topic that is DNA replication. So DNA, that is a double helical structure which is discovered by Watson and Crick, and this DNA helical structure, it is also called as a Watson and Crick model. So Watson and Crick developed their ideas about the genetic replication in a second article in Nature, and which was published on May 30th, 1953. The two had shown that in DNA, form is a function, that is, the double-stranded molecule could both produce exact copies of itself and carry the genetic instructions. So this is the DNA double helical structure. So there are three types of DNA double helical structures. First one is the key features of DNA structure, in which we can see that there is a distance between two bases, two pairs of bases, that is 0.34 nanometer. The second one is a distance between two helices is 3.4 nanometer. The second structure is a partial chemical structure that shows the chemical nature of DNA and the bonds which are present between nucleotides, that is adenine and thymine. They are bound together by the 2 hydrogen bond. For example, the second one is the guanine and cytosine. They are bound to each other by the 3 hydrogen bonds. And the phosphodiester bond is present between two deoxyribonucleotides. Okay. The third one is a space filling model, which was shown by the Locken, the ball and stick model. Now, the directionality of DNA. So, DNA molecule is having particular direction. Each strand of DNA is having particular direction. Nucleotide in DNA backbone are bounded together by the particular bond, which is called as a phosphodiester bond or phosphodiester linkage, which is between 3' carbon and 5' carbon atoms. The DNA molecule has a particular direction, that is, if the one strand from two strands, one strand bearing the direction of 5' to 3' and second one is a 3' to 5'. That means the complementary DNA strands run in opposite direction. Now the DNA replication introduction. So DNA, it is the basics of inheritance. That means it carries some characteristics or some characteristics from parent to its offspring. Fundamental process occurring in all cells for copying DNA to transfer the genetic information to daughter cells. Each cell must replicate in its DNA before its division. So, proposed model of DNA replication. There are different types of proposed model of DNA replication. In the late 1950s, three different mechanisms were proposed for the DNA replication. First one is a conservative model. In conservative mode of replication, both parental strands stay together after DNA replication. In second one, semi-conservative mode of replication, the double-stranded DNA contain one parental and one daughter strand following the replication. And in third one, that is the dispersive mode of replication, parental and daughter DNA are interspersed in both strands following the replication. Okay, so these are the diagrams of model of DNA replication. The first diagram is showing the conservative model of replication, in which the parental DNA is conserved as it is, and the blue colored DNA shows the daughter strand or daughter DNA. Second one is a dispersive mode of replication, in which parental and daughter DNA are dispersed among the DNA double helical structure. And third one is a semi-conservative mode of replication, in which out of two double stranded DNA, the one strand is daughter strand and second one is a parental strand. So the semi-conservative mode of replication is important. Now the origin of replication we will see in prokaryotes. At the origin of replication we call it as a ori. So there is an association of sequences specific double-stranded DNA binding protein with a series of direct repeat DNA sequences. So at the origin of replication, there are some proteins which are sequence specific that goes and bind to the DNA replicating point or origin of replication. In E. coli, the ori C is bound by the protein, that is DNA. First, the DNA replication machinery was discovered in E. coli, and at the origin of replication or ori C, the first protein that goes and bind to the ori C, that is the DNA. A complex is formed consisting of 150 to 250 base pair of DNA and multimer of DNA binding protein. This leads to the local denaturation and unwinding of an adjacent AT-rich region of DNA. So, DNA A protein that goes and bind to the AT-rich region of the DNA, which this particular AT-rich region is present in the origin of replication sequences. This is the origin of replication. So, leading strand and lagging strand direction synthesis in 2 DNA replication fork of replication bubble. So, during the bidirectional DNA replication. So, at the origin of DNA replication, when DNA A protein binds and other protein comes and binds, there is formation of replication bubble, and after that there is a formation of replication fork. And this replication fork contains some leading strands and lagging strands in which the DNA is going to synthesized. This is the bidirectional mode of replication of DNA. So, at the origin of replication, there is a start of DNA replication, and there is presence of replication fork. There are 2 replication fork, and these 2 replication fork are also called as a Y model of DNA replication. Now, the origin of replication in eukaryotes. We have discussed about the origin of replication in prokaryotes. Now, the origin of replication in eukaryotes. Functionally similar autonomously replicating sequences, that is ARS, in short we call it as ARS, or replicator have been identified in each cell. The ARS contains somewhat degenerate 11 base pair sequences called as the origin replication element, that is ORE. The ORE binds a set of protein analogous to the DNA A protein of E. coli. So, ORE contain, or it is a set of protein which is analogous to the DNA A in case of E. coli, and ORE present in the yeast cell. So, which is collectively called as origin recognition sequences or origin recognition complexes or ORC. So, the ORE is located adjacent to an approximately 80 base pair AT-rich sequences. So, where the ORE is present, origin of replication element, that is ORE, it is located nearly or by the adjacent approximately 80 base pairs sequences that is easy to unwind. So, AT contain only two bonds and GC contain 3 bonds. So, that's why AT backbone will break easily, and this is called as a DNA unwinding element, that is DUE. Now, the primer. There is need of primer for the synthesis of DNA. So, what is primer? DNA polymerase can only add nucleotide to 3' end of a growing DNA strand. So, there is a need of starter. So, nucleotide to make a nucleotide can make a bond between 3' between that starter point and the newly added nucleotide. So, strands can be only in 5' to 3' direction. So, what this particular sentence shows, DNA is growing in 5' to 3' direction. See, 3' direction, it is the growing end at which the newly added nucleotide or new nucleotide is going to add over there. Template is read in 3' carbon to 5' direction, while the polymerization takes place in 5' to 3' direction. Okay, so the DNA or RNA, they are always synthesized in 5' to 3' direction. Now, the RNA primer. It may be as a RNA or it may be DNA, but generally in DNA synthesis there is a use of RNA primer. So, this RNA primer is synthesized by the enzyme which is called as a primase, which serve as a starter sequence for DNA polymerase 3. Only one RNA primer requires for the leading strand. RNA primer for the lagging strand depends on the number of Okazaki fragments. So, there are 2 types of fragments, that is there are 2 types of strands which are newly synthesized, that is lagging strand, and second one is a leading strand. The continuously synthesizing strand is called as a leading strand, and discontinuously synthesizing strand, it is called as a lagging strand. So, lagging strand contain number of Okazaki fragment. So, the number of RNA primer is depending on the number of Okazaki fragments. RNA primer has free 3' OH group to which the first nucleotide is bound. Okay. Now, there are different components of DNA replication. First one is a DNA polymerase. It carry out the synthesis of or deoxynucleotide polymerization. That is, it carry out the synthesis of DNA or addition of the nucleotide to the 3' end of the growing DNA strand. Second one is a helicase, which carry out the process of unwinding of DNA. Topoisomerase, which relieves the torsion strain that result from the helicase induced unwinding. When there is action of helicase, there is an unwinding of DNA, and at one end there is a formation of torsion or stress, and that stress is relieved by the topoisomerase. There are two types of topoisomerase, that is topoisomerase 1 and 2, and that cut the DNA at one point or 2 points so that there will be the release of stress. Now the third one is the RNA primer, which initiates the synthesis of RNA primer. After that, the single strand binding protein, which prevent the mature reannealing of double stranded DNA. So after that, the DNA ligase, this enzyme which seals the single strand nick between the nascent chain and the Okazaki fragments on the lagging strand. So DNA ligase, which is the enzyme which carry out or which is capable to seal the gap between the nucleotides. The next point is the steps in DNA replication. Identification of origin of replication. After that, unwinding and denaturation of double-stranded DNA to provide a single-stranded template. After that, the formation of replication fork. After that, the initiation of DNA synthesis, elongation, synthesis of leading and lagging strand, that is Okazaki fragments. After that, the removal of primer, ligation of newly synthesized DNA segment, and the last step is a reconstitution of chromatin structure. So these are the steps which are involved in the DNA replication. For each step there is a need of different proteins. Now there are different DNA polymerases. Okay, so there are three types of DNA polymerases, that is in case of prokaryotic cell, that is DNA polymerase 1, 2 and 3. So DNA polymerase having mass, that is 103 kilodalton, the turnover number is 600, number of subunits, that is only 1. So DNA polymerase is made up of only one subunit. Structural gene is Pol A only. Polymerization carry out in case of DNA polymerase 1, that is 5' to 3' direction. It is not having any exonuclease activity. It is not having exonuclease activity from 5' to 3' end, but it is having exonuclease activity from 3' to 5' end. Now the DNA polymerase 2, it is having mass, the mass of DNA polymerase 2 is 90 kilodalton, the turnover number is 30, but the number of subunits involved in the construction of DNA polymerase 2 is more than 4. Structural gene is Pol B. See, if there is only single gene, but in case of prokaryotic cell, the gene is made up of number of subunits. So that's why the number of subunits in case of DNA polymerase 2 is greater than 4. Polymerization is in 5' to 3' direction. DNA polymerase 2 is not having exonuclease activity from 5' to 3' direction, but it is having an exonuclease activity from 3' to 5' direction. Now the third one is polymerase 3. The mass of polymerase 3 is 830 kilodalton. Turnover number is 1200. The number of subunits are 10, and the structural gene is Pol C. Polymerization from 5' to 3' direction. It is also not having an exonuclease activity from 5' to 3' end, but it is having an exonuclease activity from 3' to 5' end. Thank you.